we'll discuss about the non-membranous cell organelle, that is ribosomes, which are playing very important role in protein synthesis. Both the cells, that is prokaryotic as well as eukaryotic cells, they are having these non-membranous systems, that is the ribosomes, where they are of two types according to their size. They are classified, and these are 70S and 80S types, where S, it refers to the Sedberg unit, which is sedimentation rate, means when they are undergoing ultracentrifugation, that time they get settled according to their density, and that sedimentation rate is indicated by this S, that is 70S and 80S type of ribosomes. 1 Sedberg unit is equal to 10 raised to minus 13 seconds. That is the sedimentation rate of that particular particle. Here, while forming these 70S and 80S types, here two subunits, they are joined to form the complete ribosome, which is granular structure. It is not covered over by any membrane, and these subunits, they are formed by the ribosomal proteins and ribosomal RNA, that is rRNA and ribosomal proteins. They are forming these small granules. So usually in case of eukaryotic cell, both these types of ribosomes, they are present. That is, in the cytoplasm we observe the first type, that is 80S type of ribosome, which are freely suspended in the cytoplasm. Even many times they are associated with the membranes like nuclear envelope and endoplasmic reticulum. So those free ribosomes, they are of 80S type, while the cell organelles like chloroplast, mitochondria, they also contain the ribosomes. These are of 70S type. But in case of prokaryotic cells, only and only 70S type of ribosomes are observed. So we see exactly how these ribosomes are formed, how they are assembled. So in case of prokaryotes and cell organelles of eukaryote, what we say, 70S type of ribosomes are present, which are formed by assembly of two subunits, smaller subunit and larger subunit, where in case of 70S type, what is the structural arrangement, we will see, and we will compare it with the eukaryotic 80S type of ribosome, how it is assembled. So here, in case of the 70S type of ribosome, the smaller subunit is of 30S, 30 Sedberg unit it is having, and it is composed of 16 molecules, that is 16S, 16 sedimentation rate, that is rRNA. 16S जो आहे याच्यामधला, that is by rRNA, and remaining 21 is by the ribosomal proteins. While forming the larger subunit, which is of 50S, which is formed by 23S of rRNA, 5S rRNA and 34 proteins. So together 30S smaller subunit, 50S larger subunit, that is going to form a complete ribosome in case of 70S type. While In case of eukaryotes, where we observe ATS type, here the smaller subunit is of 40S, which is formed by 18S rRNAs and 33 proteins. While the large subunit, which is of 60S, it is having 20S rRNA, 5.8S rRNA, 5S rRNA. So 28 plus 5.8 plus 5. These are all rRNA units, and 50 proteins are there, which are going to form this large subunit of 60 Sedberg unit, forming complete ribosome of 80 Sedberg unit. So these ribosomes, when they are present associated with the membrane systems, they are generally involved in the process of protein synthesis, which are transported across the membrane. For example, in case of acinar cells of pancreas, they are going to produce those digestive proteins, which are enzymes, and when they are present freely in the cytoplasm, that time they are going to combine, forming a group. We call it as polyribosomes. We have studied that in case of prokaryotic cell, that is polyribosomes. You have mentioned, you have labeled them in the bacterial cell. And those polyribosomes units, they are actively involved in the process of protein synthesis. So it is about the 70S type and 80S type of ribosomes, even the freely suspended ribosomes in the cytoplasm. They are producing those proteins which are helpful for the metabolism of the cytoplasm. For example, the hydrolytic enzymes for sugar, they are produced by those freely suspended ribosomes. So overall, what we can say, ribosomes are having key role or important role in the protein synthesis of the cell where they are present.